Hi, my name is Laura Gibbons. I'm a fifth-year PhD here at the Duke University Marine Lab. My research focuses on how communities respond to environmental change, and I do that using advances in molecular technology. My interest is using genetic and molecular methods to look at communities that are difficult to actually see. A lot of my work focuses around aquaculture and aquaculture systems, and the Duke Marine Lab has an aquaculture facility that's maintained by students and has been going for a few years now. Because it's very close, I can leave my lab and in like 20 minutes collect my sample and come back. That's a really unique opportunity here at the Marine Lab. I'm using eDNA, or environmental DNA. Everybody sheds DNA. We do, animals do, everybody sheds cells, and all of your cells have DNA. So if a fish or dolphin swims through the water at the aqua farm, it's going to be shedding cells. And so if I scoop up water, you will be able to see if something swam through. You get a larger picture of what was there using eDNA because you don't have to visually see it. When I'm out at the oyster farm every week, I'm not seeing a dolphin swim by or anything like that, but I know they're there. Taking a water sample, you can get a really wide range of organisms from it. I am filtering that water so it's only retaining the solids, which includes cells. So I've started processing some of these samples, looking at the aquaculture and also some of our reefs. And the integration of environmental DNA with third-generation sequencing is very new, and it's a really interesting intersection of molecular ideas and traditional marine work, which I think is really cool. Oyster reefs are really a biodiverse habitat, and there is a lot of restoration effort that's going on, but it doesn't tell us a lot about whether or not returning those oysters also returns other species. Whether or not that's happening in the restoration process can be informative to managers. Climate change affects both the oysters themselves and the communities around them. So if we are able to see who's there now and who was there in the past, then you can start to get an idea of what that community is going to look like 20, 30 years in the future.